Hello, everyone. Welcome to our last Water Ambassador webinar for 2022. The Water Ambassador Program is a partnership program between Martin County and the University of Florida ICUS Extension. It's Martin County's way to accomplish their stormwater education goals. As I said, this is the last of our webinars for 2022, but we hope to be back with a full new roster of presentations starting in the new calendar year. So stay updated for that. Just as a reminder, although everyone is pretty familiar with Zoom by now, we ask you to keep yourself muted and to turn your video off, especially with the storms that are happening now. Turning your video off helps with bandwidth and allows you to get sort of the best clarity. We also suggest if you want to put all of your questions or comments in the chat box, and if you need to leave at any time, just click the end leave meeting button. And if for your best viewing pleasure, look at the side-by-side mode. This will allow you to see both of your speakers and the presentation at the same time. All of our webinars are recorded and hosted on the Martin County Extension website. This is a mouthful, and we'll put it in the chat box, but if you just Google Martin County Sea Grant, you will get to the page, and all of the recordings are listed by the date as well as the topic. Just a reminder to please use the chat feature to ask any questions, and we'll be holding all of our questions until the end. So just be patient with us while we have our fantastic speaker, who is Abbey Gering. Abby is going to be talking about repurposing plastic bottle caps for nutrient removal in the Indian River Lagoon. Abby is a recent graduate of Florida Tech. She got her master's degree in chemical oceanography in December of 2021. She moved here from Illinois in 2016 and fell in love with the local environments like the Indian River Lagoon and being able to help improve lagoon conditions and work on projects such as the bottle cap nutrient removal study that she's going to be talking about today, alongside Dr. Austin Fox in Florida Tech's Marine Biogeochemistry lab. Since graduating, she's been working full-time as an environmental scientist at Applied Ecology, Inc., and that allows her to continue to work on the local environments. She's always been an advocate for environmental conservation and is thrilled to be able to pursue a career studying water quality and aiding in restoration projects to improve the health and understanding of our coastal ecosystems. In Abby's free time, she'll be somewhere around the Indian River Lagoon region with her camera, exploring nature trails, volunteering for restoration projects, paddle boarding along the IRL, and surfing. So she has spent most of her educational, professional, and her free time exploring our water and our water bodies. So we're super excited to have Abby here today. Vincent and I both saw her give this presentation at the Indian River Lagoon Symposium, and we thought it would be a fantastic representation of some of the out-of-the-box strategies for improving water quality. And we're super excited that she accepted our invitation to present for the Water Ambassadors. So with that, I'm going to stop sharing my screen and turn it over to you, Abby. Okay, also, for those of you before Abby gets started, we have asked Abby to, when she introduces herself, to turn off her camera because of bandwidth issues with the storm. So she will introduce herself and then she'll come back in video at the end for question and answer.
Thank you for the introduction, Lisa. I really appreciate it. If you couldn't tell, I do absolutely love the Indian River Lagoon. I fell in love with it the first time I drove over the causeway, and I love being able to work on it. So this research is actually an extension of my thesis research, and I graduated in December from Florida Tech with my master's degree in chemical oceanography. And so while I'm full-time at Applied Ecology as an environmental scientist, I do work part-time still at Florida Tech to help manage and run this project. And so very passionate about it. I love it, and I'm excited to talk to you more about it. And if there's folks on here that have been keeping up with the project and you helped donate plastic bottle caps, thank you so much in advance. We really wouldn't have been able to do this project without the support from the community. So with that being said, I'm going to turn off my video and get started. So I'm sure many people, especially if you're living along the lagoon, let's see if it goes forward, there we go. You're familiar with the term eutrophication. If not, it's just this fancy term that we use to describe an excess of nutrients. So there's too much nitrogen and there's too much phosphorus. Normally, nitrogen, phosphorus, it's not a problem. All living organisms need it, you and I need it. But like most things in excess, this is when we can start to see a lot of problems, specifically with our algae blooms. So when you have a lot of human input of nitrogen and phosphorus into the lagoon, you can start to fuel algae blooms. And so these pictures were taken more up here in the northern lagoon, the Cocoa Beach area, just last year. And so the water is matching the color of the trees. This is something we do not want to see. Not only do these algae blooms block sunlight from reaching your submerged aquatic vegetation, but also it influences the dissolved oxygen concentrations a lot. And so in the beginning, you might see in an algae bloom really high concentrations of dissolved oxygen at the surface. This is because they're photosynthetic organisms. They're using the sunlight to photosynthesize and produce oxygen. However, at night, there's no sunlight. They cannot photosynthesize. These algae are still breathing. They're using water from the water column. And then eventually they're going to die and decompose and increase the organic matter load to the lagoon. In this decomposition process, you have bacteria that are working really hard to break down this matter. And again, in this process, they are continuing to breathe. They're using a lot of the oxygen from the water column. And in very serious instances, we can actually see low dissolved oxygen concentrations below 2 milligrams per liter, and we call this hypoxia. And then in extreme conditions, you can actually have anoxia, the complete absence of oxygen. When you have low dissolved oxygen concentrations, you start to see the accumulation of hydrogen sulfide. And so if you've ever driven past the lagoon and your nose kind of goes up a little bit because it smells like rotten eggs, it's likely that you're smelling hydrogen sulfide. And so we see this a lot in low oxygen environments. And not only is it not a pleasant smell, but it also is toxic for things like seagrasses. And so a lot of people are aware of the drastic loss of seagrasses the lagoon is experiencing, as well as with the manatees. And that's kind of where the conversation stops. So I want to take it one step further and ask you, why are the seagrasses dying? So the seagrass are dying because the sediments are dying. And I know this might sound silly to think of. I want a big sign that says SOS, save our sand. Because believe it or not, we are losing healthy, viable sandy sediments. And this is a really important component of our ecosystems. Throughout the lagoon, we're seeing this accumulation of really fine grain, organic, rich material that's locally called muck. And let me just tell you, be grateful you cannot smell this picture. It is not pleasant. So this muck kind of coats the bottom of our lagoon and it's creating these really hostile environments. So it's suffocating our seagrasses. It has the accumulation of hydrogen sulfide, and it really just creates these hostile environments that impact your biogeochemical processes and ultimately the overall water quality of the lagoon. Ultimately, our goal with this project is to really understand these natural processes happening within the estuarine sediments and the water column, so we can work to promote ecosystem services through various restoration efforts. And I'll talk a little bit more about some of the research being done at Florida Tech to address these problems as well. So I'm going to try to take it easy on the geochemical aspect side. It gets a little complex, but really the main thing that you need to know is there's natural biological processes known as nitrification and denitrification. Don't get overwhelmed by this image. All you need to pay attention to is the top layer. So you see where it says oxic. And healthy sandy sediments, so like the picture in the background, you could take a dissolved oxygen probe, stick it in the first couple millimeters, centimeters of the sediment, and you'll see that there's oxygen present. We call this the oxic layer of the sediment. This is a really important area. In this oxic sediment, this is where you have nitrification. So you have specific groups of bacteria, nitrifying bacteria. They're able to oxidize different forms of nitrogen like ammonium to nitrite and to nitrate. And from here, a different group of bacteria, heterotrophic denitrifying bacteria, they can then reduce this nitrate to nitrite and eventually to N2 gas. And we want it to go to N2 gas because then it'll diffuse through the water column into the atmosphere, which is 78% nitrogen. So again, these are completely natural biological processes. They're constantly happening in the water column, but also predominantly in the sediments. And so you can see where this denitrification occurs, it's in that anoxic layer. So I mentioned earlier, anoxic is devoid of oxygen. There's no oxygen present. And that interface between where that oxic and anoxic sediment meets is extremely important for these biogeochemical processes. Sediment denitrification, it's supposed to be responsible for removing a large fraction of the external nitrogen that we are introducing to the system. And I'll get in a little bit later why we're not seeing this so much. And because they occur in this oxic-anoxic interface, when we see the accumulation of something like muck, we're losing this healthy, viable, sandy sediment for these processes to take place. If you were to stick the same dissolved oxygen probe into the first millimeter or centimeter of muck, it would be completely devoid of oxygen. You would not have that oxic layer. And it seems really tedious, but it is. They're really complex processes. And so that one little layer is extremely important. So why aren't we seeing these processes more in the estuary? And this is what we're really trying to understand with this project, because if we can understand how these ecosystem services work, it's in the environment's natural buffer against our human impacts, we can help promote them and kind of help stabilize the system more. So one reason we're not seeing this is because it's variable environmental conditions, especially with the lagoon. These biogeochemical processes are going to be strongly impacted by things like temperature, oxygen, salinity, pH, whether or not we have that accumulation of toxic hydrogen sulfide. All of these things play a part in these processes. All of these things are really important, but unfortunately in the lagoon, they're always changing. And so one side of the lagoon could be completely different from the other. The surface of the lagoon can be completely different from the bottom. For example, this is some data from Dr. Austin Fox in the biogeochemistry lab at Florida Tech. He is growing this network of bottom water dissolved oxygen sensors. And what I really want to point out here is you can see this green line. This is where the oxygen sensor is, right above sandy, healthy sandy sediments. And so you can kind of see this up and down pattern. We call that diurnal changes. So during the day, you have your photosynthetic organisms. They're happy, they're thriving off the sunlight, they're able to produce oxygen. So we see this peak during the day of oxygen. And then at night, there's no sunlight, they can no longer photosynthesize, but these organisms continue respiring. They're still using oxygen from the water column. And so you see these peaks at night. If you look at that blue line, this is dissolved oxygen right over muck. And so it's going hypoxic, so less than 2 milligrams per liter, a lot more than the sandy sediments. And so again, the muck is that hostile environment. There's a lot of decomposition occurring. So a lot of bacteria working to break down all of this organic matter that's being added to the system. In doing so, can create more frequent hypoxic events. And when you lose dissolved oxygen at the bottom, you're changing all of those biogeochemical processes in the sediments. Another thing is that salt inhibits nitrification. So nitrification is a really important step in these processes because this is what will fuel denitrification, which is the ultimate nitrogen removal step. With salt, depending on whether at the inlet you're more north, your salinity is going to change throughout the lagoon. We're also degrading the habitat. So I talked a lot about the muck, and when you have eutrophication, you have increased organic matter loading, you're impeding on the nitrification process, which then limits the denitrification, so the overall nitrogen removal. With hypoxia and anoxia, you're decreasing the efficiency and role of nitrification and denitrification as a sink for nitrogen. So when I say sink, you can kind of like think of it as a sponge, something that'll take up nitrogen, take up phosphorus, and hold on to it. Now, if you were to flip it and you have hypoxia at the bottom water, all of a sudden, you're again changing the biogeochemical processes in the sediments, and you're actually going to release nutrients from the sediments, and it then becomes a source. And this is something we do not want to promote. When you have sediments like muck, you're eliminating that oxygen-oxic surface layer in the sediments, and you're impeding these nitrification processes. And sulfide concentrations, not only do they inhibit seagrasses, the growth of seagrasses, it also inhibits these nitrifying bacteria. So we've kind of got a lot of things stacked up against us, and it's making it hard for the system to kind of balance itself out the way it naturally would. In the Fox Biogeochemistry Lab at Florida Tech, we do a lot of work with muck. And so specifically, we go around the lagoon and it's called probing, and we probe to identify muck thickness, where the hot spots are, kind of get a general idea of the composition. We'll take some samples back to the lab, analyze it for hydrogen sulfide, and help guide restoration efforts for these muck spots. And so this is Dr. Austin Fox. He's the lead scientist in the lab, and at the specific spot, again, I'm really happy that you can't smell this picture. I'm having flashbacks, but you can see that this specific muck deposit was well over 6 feet deep. It was very clay-like, very organic rich, very, very, very smelly. And this is just an incredibly hostile environment for these biogeochemical processes. We can also measure nutrient fluxes. So I mentioned that sometimes healthy sediments can actually be a sink for nutrients. But if you have something like low dissolved oxygen events, or you have hypoxia, it can actually be a source of nutrients. And so we deploy these benthic chambers over these muck deposits, and it's really just this controlled environment. We have a chamber equipped with sensors. We're able to measure the dissolved oxygen. We take nutrient samples in the beginning and at the end to see what is changing, what is happening at that sediment water interface, how are the sediments responding to low oxygen. All of these different aspects come into play. One thing that we've learned from this research is that virtually all of the nitrogen that's being released from these muck deposits is in the form of ammonium. And this form of nitrogen specifically promotes harmful algae bloom species like Oreombra. And so these are the big blooms that we were having 2011 that kind of caused the shift in the lagoon regime. It can also do the same thing for phosphorus, and it can act as a source of phosphorus in these situations with low dissolved oxygen concentrations. So Dr. Fox has been working really hard to establish his bottom water dissolved oxygen network, and also just understanding the impacts of aeration. So this is kind of where this project really stemmed from. Was we were looking at if adding an aerator to canals would help improve the water quality in these areas, and we found limited success, and we started learning it's because of these processes like nitrification, denitrification, and we're having too hostile of environments for them to take place. So with this project, starting to get into it, I'm not going to get too much into Animox. This is another nutrient removal process. It's kind of more new in the science world. Still a lot of work being looked into, but we're just going to focus on denitrification and nitrification. And so these processes have already been engineered. They're natural, again, they're natural processes. And they've already been engineered by existing systems. So for example, wastewater treatment facilities, recirculating aquaculture systems, large marine aquaria, they all utilize these processes to help keep the nutrients down in the systems. The very beginning of my thesis, it feels like ages ago, I tested a bunch of these different denitrification media, and a lot of these systems will use this media to promote the growth and proliferation of your nitrifying and denitrifying bacteria. Specifically, nitrifying bacteria, they're extremely tedious. They are high maintenance, they're slow growing, they require very specific conditions, and they are easily impacted by changes in the environment. And so by providing them a media, it's kind of more of a hard surface that they can grow on. You'll get this really slimy little biofilm growing, and that's what we like to see. And so I tested these different media going across the screen from left to right. The first one being plastic bioballs. These are typically used, your home aquarium might have it. Large aquariums definitely use them. And then we have biochar, sand, zeolite, perlite, corn, or wood chips, and corn maize. And so I tested these all in laboratory experiments to look at the removal efficiency. So once the bacterial communities become established, we were looking at the nutrient removal capabilities. And I found that the bioballs all the way to the left, that plastic media, they were yielding really promising results. Originally with this project, the goal, we were working with the dredge projects to treat the effluent. So you suck the muck out of the water, but then what happens? You need to treat that water because it's high with nutrients. And so that's kind of where this project stemmed from. And when you're doing research and you're doing lab experiments, something that you always want to keep in your mind is this feasible? Can I scale this up to have a broader impact? And so once we were looking at this, we realized that the cost of denitrification media was quite expensive. And so we wanted to look for a more environmentally friendly and also cost-effective approach to media. And so cue the plastic bottle caps. We chose bottle caps because they're made out of high-density polyethylene and polypropylene. This is the same material as your commercially available plastic media. They're also BPA-free and they're readily available globally. And so this was the most appealing aspect of using bottle caps. I'm sure after this presentation, you're going to start to notice bottle caps everywhere. I mean, you walk across the parking lot to a store, you will find them on the ground. You walk on the beach, you'll find them on the sand. They are virtually everywhere. And so if you go to your underrepresented and underserved communities, these are the areas that have poor water quality. But these treatment systems are usually very costly, and it's hard to kind of make something that can be applied in these areas. And so our goal in the future is to create these systems as inexpensively as we can, and hopefully get them out to the areas that need them the most. We very much like this idea rather than the idea of increasing your plastic production. This is something we definitely would like to avoid. Into the science of it a little more, when I say specific surface area, that's just how much space is there available for these bacteria to grow. And so all those little grooves in the bottle cap, they help expand this surface area. And so we measured on average up to 400 meters squared per cubic meter versus your commercially available media that's up to 500 meters squared per cubic meter. And so for something that's free, it's readily available, we're giving it a new life, we're repurposing it. We thought this was a lot more beneficial than increasing our plastic production. And so right now, the stages that we're at, we have laboratory bioreactors that we have set up in the lab to help optimize our field system. And so it's a really simple concept. We take influent water from these tributaries, we pump it into a reactor cell. This is where we have the repurposed plastic bottle caps. We can control the residence times. When I say residence time, that's really just how long is this water in contact with this bacteria, with these bottle caps. And then we're creating the right environmental conditions through a lot of literature reviews and understanding what environmental conditions can promote the growth of these bacteria. We also have an additional cell in case we want to experiment with longer residence time. If all of a sudden we start treating really high concentrations of nutrients, we can kind of switch it up and do as we please. And we also make sure that our bacteria are happy and have plenty of oxygen. Getting into the field aspect of this project. So we wanted to scale it up and increase our impact. So thanks to the Indian River Lagoon National Estuary Program, we have had this pilot study funded. It's just wrapping up its first year. Treating stormwater from a local tributary up here in Melbourne. Thanks to the community support, let me just tell you, this is a sliver of the bottle caps that we have. We have well over 1.5 million, and we have put them into this tank and we have filled it up very easily. So thank you to all the community support for this. All of these bottle caps were collected through donations, people finding them on the beach, and keeping track of them and sending them our way. So the way this system works, it's very similar to the laboratory systems. Really, we're just taking influent water from our local tributary, pumping it into our Oxsolve system. And so we have been partnered with Lapin Services throughout the duration of the study. And they were even with us when we were looking at aeration studies. And so they have been incredibly supportive of this project, and we use their Oxsolve system to make our bacteria happy and give them plenty of dissolved oxygen. And so we pump water into the Oxsolve system, it's aerated, and then it's pumped into the bottom of our bioreactor. The treated effluent water slowly trickles out the top of the system, and we monitor it weekly. We take samples of that influent water that's coming in, we take samples after it's been treated, and we analyze those in the lab. Something that I'm sure a lot of people are curious about, it's like, is this girl nuts. She's using plastics. We want to avoid plastics. I absolutely agree. So we have been carefully monitoring microplastics to ensure that we are not adding to the problem, to make sure that our bottle caps are not breaking down, that we're not sending them into the environment. And we know that the two main variables that contribute to secondary microplastics is your ultraviolet radiation, so those strong Florida rays, and physical forces like wind and waves. So if you're walking on the beach and you see these tiny little, really frail looking microplastics, that's a result of just intense sunlight. You have the salt water breaking them down, you have waves crashing them to shore and breaking down smaller pieces. But in our bioreactor, as you could see in the previous pictures, that it's completely not exposed to sunlight. All of the bottle caps are within the treatment system. They have 2 layers of confinement. So for example, if we have something like a big hurricane coming through and we're worried about strong wind and waves crashing up against our system, if something were to occur, we have a secondary containment system. It's also very low energy, so there's not movement really. The caps aren't being jostled around. There's nothing in there physically breaking them down. And to just make sure of all of this completely, I've been following NOAA standard procedure for microplastic counts, and I filter the water that's coming in, filter the water that's leaving, and then I get to work with the microscope and I count each filter to monitor the microplastics. So getting into the results, looking at this graph, we're looking at inorganic nitrogen. When I say inorganic nitrogen, this is comprised of NOx, which is nitrate and nitrite, and ammonium. Looking at the graph, this is what's coming into our system. So pumping this in from the tributary, it's relatively low nutrient concentrations compared to early on in my thesis when I was treating dredge material, we had concentrations up to 90 milligrams per liter of nitrogen. So this is a lot lower concentrations, but regardless, we want to try to minimize any nutrients that we can coming into the lagoon. And so you'll see these little orange marks. Those are days we kept very detailed notes and a track record of how the system was performing. So if we had something go wrong with our oxygen, if we had something go on, we feed the bacteria, we give them a carbon source. If that pump malfunctioned, we want to keep track of all of these things, but we still want to sample. We still want to see how these flaws will impact the overall efficiency of the system. So that's what those little orange days are. And you can see after about an 8-hour residence time of treatment, obviously those days where it was malfunctioning, the system didn't perform to its expectation, and that's fine. We're learning. We are taking what we can learn. So we know, okay, if all of a sudden we run out of feed for the bacteria, the system might not perform as well, but you can see right after it bounced back really quickly. Even with these days where it wasn't fully functioning, we're still seeing on average a 72% decrease in ammonium concentrations and an 82% decrease in nitrate and nitrite concentrations. Looking at phosphate, again, those brown days are the days where we knew that there was something going wrong in the system. We made note of that so we can explain these different results. Despite those days, again, we're still seeing a 70% decrease in phosphate concentrations. This study has been funded for another year, so we're using all of this data, working in the lab systems, using our field data, and trying to continue to improve this bioreactor to remove nitrogen and phosphorus from the lagoon and from stormwater coming into the lagoon. Now, one thing that was really exciting to see is that we're actually seeing a significant difference in the count of microplastics per liter coming into the system versus what is leaving the system. And so keep in mind, all of this is under a microscope. These are really, really, really tiny, small little pieces of plastic. A majority of them were little fibers, like from your clothes, from rope, fishing line. Those are the most dominant plastics that we're seeing. Thanks to research done by people like Holly Sweat, she actually found that when you grow biofilms, so that's what we're doing, we're growing these biofilms on the plastic bottle caps, but when you grow them on microplastics, it's going to cause these microplastics to sink. So I mentioned that we have a very low energy system, and so what we believe is happening is that you have this biofilm growth on these microplastics coming into the system, and it's actually causing them to sink and settle down at the bottom of our system. So we're actually significantly able to remove microplastics from the water with this system. And to date, it's been a full year of using the same bottle caps. We have yet to find any fragments of plastics leaving the system. So there's no indication that these bottle caps are breaking down at all. And we're removing microplastics, so that was really exciting to see. An additional associated benefit that we also were not expecting was improved turbidity. And so although water clarity doesn't necessarily mean water quality, you can imagine that this would be beneficial for things like our submerged aquatic vegetation. So our seagrasses, they need that sunlight, they need clearer water to have that sunlight penetrate through the water column. To date, there is a significant difference between influent turbidity and effluent turbidity, with 5.6 NTU in the influent water and 3.2 in the effluent water. And so we are seeing improvement in water clarity. We were not expecting this at all, so it makes us really excited to see this. But one of my favorite parts about this project has been the potential for outreach. And so we have stopped accepting bottle cap donations. We have plenty for our next funded project and to fill up another bioreactor. So we've paused the collection, but the outreach has not stopped. And so if you do reach out to me through my Florida Tech email, again, this is my kind of part-time job, so I'll get back to you as soon as I can, but you'll likely see this bottle cap flyer. And so I think it's really important that the community stays up to date with the progress of this project, because again, we wouldn't have been able to do this without the community. And so throughout the duration of my thesis research and even since graduating, I've been able to have these conversations with the communities, talk to schools and help raise awareness about the Indian River Lagoon, what's going on with it, environmental research happening at Florida Tech, and also different restoration projects. And so on this flyer, not only are we updating people on the results of this project, you know, here's where your bottle caps ended up, this is what they're doing, this is how much nutrients they're removing, but I also like to provide links to other local volunteer opportunities like Marine Resource Council, Brevard Zoo, Save Our Indian River Lagoon, Brevard County, the IRL-NEP, all of these places, we're all on the same team. We all want to help. We all want to partake in restoration efforts. And so you can click on these links and find out ways that you can help in your local area. And also just kind of what you can do as a person in your household to help improve water quality in the lagoon. I will never stop believing in people's potential to make a positive change. I absolutely think one person is capable of making a change, because if you have one person with that mindset, you are going to have a butterfly effect. And the next thing you know, your community has that mindset. We come together and we can make the change. So never think that your impact is insignificant, because I promise you, everything that you do has an impact. You can choose to make it a positive one. Also, my favorite part was some of these schools went above and beyond with their collection efforts. And so I got sent pictures of these amazing displays that people made. Kids were coming home and educating their parents. I had someone email me. He said that his 7-year-old son, you know, ran up to him with this flyer when he came back from school, and he told them how excited he was. And the dad, you know, was like, well, what do you get out of this, you know, for collecting these bottle caps? And the 7-year-old responded to him, well, Dad, we get a clean river. And so seeing emails like that, it definitely makes it easier to keep pushing through. And this was just a handful of the schools that became involved, whether it was me giving presentations to them, or they were setting up these amazing collection sites, reaching out for material that they can bring into the classroom. The list goes on. And so this was a really great opportunity to improve our outreach and communication, because again, you don't really hear people often saying, save our sand. And I'll never stop saying that, save our sand, because when you think about it, sediments are the basis of our ecosystem. All of these tiny microbial processes are taking place, but they're so important, and it's such an important part of our ecosystem. So I think it's really important to learn about these lesser known ecosystem services like nitrification and denitrification. I think outreach and education is the key for lagoon restoration. If people don't know, change it, have a conversation with them, let them know what's going on in the lagoon right in their backyard. It will make the biggest difference. And so that was my favorite aspect of this project, was just all of the outreach that we got to do. I was in the classrooms, as well as, you know, more COVID time, so a lot of it was virtual, but seeing the kids being excited to contribute, seeing retirement communities excited to contribute, it was absolutely heartwarming. And so it was my favorite part of this project. I love getting to have these talks. I love talking to the community about this. And I could not be more appreciative of everybody's help. And so this is an all hands on deck project. A huge thank you to the IRL NEP. This was our first like big funded project. And so to have them believe in us and to have them funded again for second year, we're really excited to keep learning about these ecosystem services and helping other people that are participating in restoration efforts kind of guide them to how they can promote these ecosystem services in the lagoon again. Tyler with Brevard Zoo, he has been extremely supportive, and just my advisor, Dr. Austin Fox, he cares so much about the research that he's doing. He loves this lagoon with every ounce in him, and all of these people coming together, I think we're on the right direction to restoring it. So if you have any questions, comments, concerns, I know it was a lot of information to digest. These processes are kind of complex, but they're really important nevertheless. We've been a little slow on updating our Facebook page, but we're going to get back into it and hopefully have some updates in there soon. You can always reach out to me and contact me through my Florida Tech email. It's kind of easier to keep these separate from my full-time job. And so if anyone has any questions, now will be the time to ask.
Great, thank you, Abby. Fantastic presentation, and we do have lots of questions stacking up in the chat box. So while we go through the questions, I'm going to launch the quick evaluation. Shouldn't take you more than 20 seconds to answer, and we appreciate your responses. So the first question comes from Vincent. He asks whether or not the bottle caps reach a saturation point in their nitrification, denitrification activity, and do they need to be refreshed or replaced?
So something, another media that worked really well was the biochar, but that was kind of an associated problem with biochar, is there is absorption capacity because it's the biochar itself that's helping kind of absorb the nutrients. With the bottle caps and plastic media, you're actually really just growing the bacterial community on the cap itself. So it's not actually taking anything in to reach its like carrying capacity. And so we've used these caps throughout the duration of the project, and we didn't see a breakdown of them, and we saw the same performance throughout.
Great, thank you.
Mm-hmm.
Tess asks whether or not there are measurements to account for the polymer's chemical makeup that may leach harmful toxins with time.
So we have talked, we've talked a little bit with some folks that are studying plastics at FWC, and so very beginning, obviously when people hear that we're using plastics, rightfully so, there's a red flag kind of raised. So we had a lot of these meetings at the beginning with different entities, asking them questions. And the reason that we stuck to the plastic bottle caps was because they're BPA free, and since it's a food grade plastic, you are less likely to have these influences. We are in the future hoping to, we're looking for a lab right now to help. There's recent 2022 articles coming out that the reason, if you think about the why plastics are a problem in the first place, it's because they sorb anything that is hydrophobic, anything that doesn't like water can sorb to them. So we're actually trying to use this to our benefit because we have this confined system. So, you know, if we put it in a boat marina, can we actually sorb some of the oils and help remove it and send it off to hazardous waste? And so those are things that we'd like to look into more as we continue this project.
Thank you. Pat asks whether or not the muck has been tested in Lake O.
Our lab specifically have not done a lot of muck work in Lake Okeechobee. We are predominantly more up in the northern lagoon, central lagoon area.
Yeah,
I'd like to do work at the lake.
there is some work going on in sediments and sort of just the nutrient capacity of sediments, including muck in the lake.
Yeah, so Jordan Beckler at Harbor Branch, he does a lot of work down there and on Lake Okeechobee, so he'd be your go-to guy for that.
You may have answered this in response to Vincent's question, but Marion asks, do you clean and reuse the bottle caps, and what do you do with the bottle caps once they're no longer useful?
So we've been able to continue to use the bottle caps. We've not had to dispose of them, and the project keeps getting funded. So we keep having systems to supply these bottle caps to. In the future though, if we come across this problem, there's local groups that we've been looking into, like the Lions Club. Some of them actually do their own bottle cap collections. I think it kind of got paused with COVID, but they were actually collecting bottle caps to send to Indiana, and they were being repurposed and made into benches. And so we've already been looking into how can we give this another life after this? It's a great question.
Sharon asks about the specific test used for microplastics.
So we've just been following NOAA's standard procedure. They have a whole flyer that's available online, step by step, what procedures to take. We do it all in a clean room, in a clean environment, obviously not wearing clothes where you'd have a lot of fibers coming off. And then we filter what is coming into the system on little gridded 0.45 micron grids. And then I go to work with the microscope and I just count each individual grid and count all the microplastics on there.
Vincent has the same question I had, which is about the capacity of these bioreactors. So he asks, what's the average volume of water that can be treated by this size system? He's wondering if this can be applied to small canals.
So that's actually our next, we were just awarded DEP funding. So kind of our next step, rather than scaling it up more, we actually want to scale it down a step and see if we can create mini systems for, for example, if people wanted it in their canals. But again, we're learning as we treat different water bodies, there's going to be, the whole point is that we're taking in natural bacteria that are already present in the lagoon. We're not growing them in a lab. We're not buying them and putting them into the system. We're just kind of taking what's already there in the lagoon, giving it the environment that it wants and it needs, and then seeing it grow from there. So one of the things that we're going to have to test and kind of overcome is if we want to apply these in different systems, we're going to have to get a good understanding of what kind of the general best environmental conditions could be used for a majority of these locations. And so I'm excited to keep working on this project. That's why I wanted to stay on it. There's a lot of different paths that we can go down. And I think there's a lot of room for always improving it and learning. And that's my favorite part about science.
Thank you. And we have a bunch of questions still to be answered. But in the meantime, while we go through those, we are accepting requests. If there's a presentation or a topic that you're interested in for the next Water Ambassador webinar series, throw it in the chat box and Vincent and I will take note of that as we plan our schedule for 2023. Pat is interested in contributing bottle caps. Their 55 and older community would love to find a source for already saved caps. Are you still accepting donations?
We are not at the moment accepting donations. We have probably now like over 2 million caps. And so we want to make sure that we're able to use everything that we already have. And it's a big sorting process. And I see the question following that is asking about like discarded pill bottles and stuff. And so a lot of the times we'll get big bags of caps and we actually have to sort through them by hand and pick out only the bottle caps, because again, we know that the bottle caps are BPA free and it's a food grade plastic. So we feel safe putting this into water. Something like discarded pill bottle caps, I would not want to use because you don't know what could leach off of those.
And Vincent asks whether or not solar power could be used to pump the pumps that move the water into the bioreactors.
So again, this is what we're looking at for the next round of funding to see for the little like canal systems, if we can find an affordable. So the only thing with solar is it kind of increases the price a little bit depending on what you go with. So we're trying to still look into more cost-effective approaches to keep the price down so we can apply these in your underrepresented and underserved communities. But I love the solar panel idea.
Do you have a sort of a vague range of what these reactors do cost now, knowing that they're not optimized?
I actually do not have a cost of that yet. We haven't really gotten to that far. We have like a little prototype that we've started working on once the funding starts up. But hopefully, as we learn more, I keep updating our flyer that I distribute to people that reach out to me through email. So I'll keep in mind, I'll keep doing my research and looking for any other organizations right now accepting bottle caps, and I'll add that to that flyer. That's a good idea. So thank you, Pat. And I'll keep kind of adding on to this information to keep the community updated.
Great. And following up on the repurposing of the used bottle caps, Pat says that the local buddy bench projects ended because unfortunately it looks like the plant burned down or went out of business or something. So
that's so unfortunate.
Yeah.
Well, I'll have to up my research and find some more groups that can. I know there's some local artists even that use bottle caps to create art. So that's kind of the next direction of paths I'm going to try to look at and target some people to see if they need any of those caps.
And I agree with Vincent. Great presentation. Thank you so much.
You.
And it's super exciting for us to be able to end on a positive note. You know, unfortunately, many of our water quality talks can be depressing, but I think this is a super encouraging and positive presentation, and we're really glad to be able to end on a high note. So thank you again for a fantastic presentation.
Yeah, of course.
Vincent posted the latest Hurricane Ian update. Stay safe, everyone. And as hurricane and potential for flash flooding does come into local communities, keep in mind the potential water quality impacts. So since this is a stormwater webinar series, don't fertilize your lawns now. It's a terrible idea.
Yes, thank you. Yeah,
clear out any storm drains of litter or debris to help sort of remove the organics from going down there, but also minimize potential for flooding. And the worst case scenario, if you have any chemicals in your garage or something, elevate them so that they don't spill or float away.
Yeah, please be safe, everybody.
Yes, please be safe. And thank you again for joining us for the 2022 Water Ambassador webinar series. And we look forward to seeing you all in 2023. Thanks, everyone. And thank you, Abby.
Bye.
Thank you, everybody.